The title of today's presentation is Chromatographic Techniques as Applied in Microbial Metabolite Analysis. The purpose of the presentation is to complement the physical classroom lectures so as to serve as a reference for the student. Now, introduction. Microorganisms generally produce diverse range of primary and secondary metabolites with applications in medicine, for example, antibiotics, agriculture, metabolites such as biopesticides, and industries, for example, enzymes, pigment, and so on. Now, accurate analysis and identification of this metabolite requires advanced analytical tools, especially chromatographic techniques, which is the focus of this presentation. Now, in this presentation, we are going to focus on thin layer chromatography, high performance liquid chromatography, and gas chromatography, mass spectrometry. Taking the first technique, which is thin layer chromatography. This technique is one of the simplest, rapid, and inexpensive chromatographic technique that is used to separate non-volatile mixtures over a solid adsorbent stationary phase, usually aided by a liquid solvent called mobile phase that moves the analyte through the stationary phase by capillary action, leading to separation of the component along the stationary phase. This technique is applied it has several applications, such as in preliminary screening of microbial secondary metabolites. It is also used in the identification of antimicrobial compounds, pigments, and secondary metabolites. It is also used in bioautography to detect bioactive compounds. For example, it can be used to detect secondary metabolite such as actinomycin D, which is produced by Streptomyces species. For this technique, the first step involves sample preparation. During sample preparation, the extract of the crude metabolite is obtained using solvents such as methanol, ethyl acetate, and so on. And subsequently, the extract is concentrated and dissolved in a minimal solvent for spotting on the TLC plate. The next step involves sample spotting, development, and viewing. In this case, a spot of 5 to 10 microliter of the extract is made on the TLC plate and subsequently developed in a TLC chamber containing the mobile phase. During the development, the mobile phase moves along the stationary phase together with the analyte through the process of capillary action. Now, during this movement, the components are being separated from each other based on their relative sizes and affinity to the stationary phase. After development, the samples are viewed under UV light or using by application of some reagents such as hydrogen sulfides to visualize the TLC spots. Now, thin layer chromatographic technique can be used for both qualitative or quantitative purpose. If it is used for qualitative purpose, it is called qualitative thin layer chromatography. This technique is used to detect the presence of specific analyte in a sample. And to do this, a commercial standard of the target analyte is spotted and partitioned alongside the test sample. And the bands produced by the standard are compared with that of the samples to confirm the presence of the analyte in the various samples. In other words, the retention factor of the standard is used to confirm the presence of the analyte in the various samples. Quantitative thin layer chromatography, on the other hand, is used to detect and quantify analyte in a sample. In this case, a serial dilution of a known standard is spotted on TLC plate, developed, viewed, and scanned with an image scanner. Now, the obtained TLC image is then imported into a TLC quantifying software such as just TLC software to determine the densities of the spots of the respective TLC spots. Now, the densities of the respective TLC spots will then be used to plot a calibration curve of the densities versus the respective densities versus respective concentration of the standard. Now, through the calibration curve, an equation will now be obtained, which is the calibration curve equation that can now be applied to determine the concentration of the unknown sample. Like in this example, standard aflatoxins are spotted. After spotting, the samples were developed. After developing, the image was then imported into a just TLC software to quantify the respective densities. After quantifying the respective densities, a calibration curve is then constructed, which is the concentration of the spot versus the relative densities. And based on that, equations were produced for each of the aflatoxins. Aflatoxin B1, aflatoxin B2, aflatoxin G1, and aflatoxin G2. Now, in this case, to determine the concentration of an unknown, the unknown analyte will then be analyzed using the same solvent system, and then the image will be imported into the TLC spot quantifier to quantify the density, and then the relative density can now be substituted in the equation to determine the respective concentration of each aflatoxin. These techniques has several applications. For example, it is used in tracking the progress of chemical reactions by observing the differences of the reactant and appearance of the new products. It is also used for compound identification through comparing the RF values of the unknown compound with the known standard. It can also be applied for purity assessment to determine the number of components in a mixture and their relative concentrations. It can also be used for sample purification to separate and isolate a small amount of compounds for further analysis. It can also be used for food and cosmetic analysis to quantify and identify food coloring agent, preservatives, or other additives. It is also used in environmental analysis to detect pollutant and contaminant in environmental samples. It can also be used in biochemical analysis to separate and identify biochemical compounds, biomolecules such as lipids, sugars, amino acids, and so on. In fact, it can also be used to provide a fingerprint of compounds in a particular analyte. Now, let us quickly look at the process of developing a quantitative thin layer chromatography. Generally, there are 3 steps involved in development of quantitative thin layer chromatography. The first step is the mobile phase development, followed by validation, and then performance evaluation. Now, for mobile phase development, a standard analyte is partitioned using several solvent systems until a good separation is achieved. For example, in this plate, line A A, B, C, D, E, F, and G represent a mobile phase development system for aflatoxins in a sample using several solvents. Line A is the solvent system used is acetonitrile and dichloromethane in a ratio of 1:10. Line B, acetone and dichloromethane was used in a ratio of 3:30. Line C, acetone and dichloromethane was used in another ratio. Line D, E, F, G, and so on. Now looking at the plate, the best solvent system that produce a good separation of the aflatoxin is the solvent system used in lane F. Looking at it, the aflatoxins were well separated, although lane D and lane E also produce a kind of separation of the aflatoxins, but the solvent system used in lane F has a very good polarity that produce better separation of the aflatoxins. Hence, the solvent system in lane F will now can now be adopted for analysis of aflatoxins, or if it is not aflatoxins, it can be adopted for the analysis of the analyte, the target analyte. However, this solvent system has to be validated. It has to be subjected to further validation before being adopted for analysis of unknown samples. In other words, the developed TLC must be validated to ensure it meets up with scientific standard benchmark before it can be put to use. During the validation process, several parameters are measured, such as linearity of the method, sensitivity of the method, specificity of the method, accuracy of the method, precision of the method, robustness, and so on. Now, let us look at the first parameter, which is linearity. Now, linearity refers to the ability of an analytical method to produce test results that are directly proportional to the concentration of analyte in the sample within a given range. In simpler terms, as the amount of the analyte increases, the response such as the density of the TLC spot increases in a straight line, linear manner. Now, in order to determine the linearity of a method, usually is assessed by preparing and analyzing a series of standards at different concentration, and then constructing a calibration curve to confirm the response versus concentration relationship in a straight line. Now, looking at this example, the standards were serial dilution of the standard were measured, and then the calibration curve is produced, and then from the calibration curve we have the calibration curve equation, and we have the coefficient of the calibration curve, that is the correlation coefficient or R-square value. For example, in this figure, 4 aflatoxins were analyzed: aflatoxin A, B1, B2, G1, and G2, and then the respective equations were produced based on the calibration curve equation, and the respective R-square values were obtained. Like for aflatoxin B1, the R-square value is 0.9974, aflatoxin B2 0.9949, and so on. Now, a good linearity of the method must be able to produce an R-square value of greater than 95%, that is 0.95. R-square values, linearity with R-square values less than 0.95% usually are not considered very good. Now, the second aspect that is being assessed during the validation steps is the sensitivity of the method. That is limit of detection and limit of quantitation. Determination of the limit of detection and quantitation of the method. Sensitivity of analytical method refers to its ability to detect and measure small amount or low concentration of an analyte in a sample. In other words, it reflects how well the method can distinguish between small differences in an analyte. And that is why it is expressed in terms of the limit of detection, which is the smallest amount of analyte that can be reliably detected but not necessarily quantified, and limit of quantitation, which is a small amount of the analyte that can be quantitatively measured with acceptable precision and accuracy. So a more sensitive method has lower limit of detection and lower limit of quantitation values. These parameters are calculated from the calibration curve equation using a formula. Limit of detection is equal to 3 times standard deviations of the response divided by slope of the calibration curve. The LOQ, on the other hand, is equal to 10 times the standard deviation of the responses divided by the slope of the calibration curve. Now, the next parameter that has to be evaluated during the validation process is the precision of the method. Precision of a method usually refers to the closeness of agreement, that is, degree of scatter between series of measurements of the same sample under specified conditions. In simpler terms, it indicates how consistent or reproducible the results are when the method is repeated. Precision is usually expressed as relative standard deviation, percentage relative standard deviation, or coefficient of variation of the measurement. Usually, precision is evaluated at 3 or 2 levels based on repeatability of the method or reproducibility of the method, or intraday precision of the method. Now, the reproducibility is defined as the variation performed by different analysts on different days or using different equipment within the same laboratory. While reproducibility is a variation that is obtained when the analysis is performed across different laboratories. High precision indicates that the method gives a very similar result each time it is applied under these various conditions. Now, the next parameter that is assessed during validation process of a method is to measure the specificity of the method. Specificity of the method is the ability of an analytical method to measure the analyze of interest accurately and unambiguously in the presence of other components that might be expected to be present in the analyte, such as impurities, degradants, or matrix component. In simpler terms, specificity is the method's capacity to distinguish the target analyte from everything else in the sample. For example, in thin layer chromatography, specificity is demonstrated if no other spot or band overlap or interfere with the analyte spot at its specific RF or retention factor value, ensuring that the measurement is solely due to the analyte. Therefore, specificity is evaluated in TLC by checking for the absence of interfering bands alongside the standard and spike samples. Now, the other parameters that are also measured during method validation include accuracy and robustness. Accuracy is usually expressed as a percentage recovery of a known amount of analyte that is added into a sample. Usually, accuracy is obtained by analyzing the recovery of samples of spike samples at 3 different levels. That is a standard or known or a known portion of the analyte is added into the sample, mixed together, and then analyzed at 3 different concentration levels, and then the recovery of the standard will now be measured from the artificially contaminated samples to obtain the accuracy. Now, the next method, the last parameter is the robustness of the method. Robustness of the method refers to the ability of an analytical method to remain unaffected by small deliberate variation in method parameters and to produce consistent, reliable result under such conditions. So in order to determine the robustness of a method, certain variations such as variations in temperature, variations in pH are deliberately done to the analyte or the mobile phase and then tested to see if a reproducible result could be obtained. A robust method should be able to produce, should not be affected by small variations in analytical conditions. Now, once a method is tested for these parameters, linearity, sensitivity, precision, repeatability, reproducibility, specificity, accuracy, and robustness, that method can be considered valid. However, some additional validations step may be added, such as comparing the performance of the method with a gold standard method. For example, in order to validate a thin layer chromatography, its performance can be compared with high performance liquid chromatography. So the last step could be the comparison between the developed method and the gold standard. For example, and this comparison is usually done by plotting a calibration curve of the concentrations measured using the developed method versus the concentration measured using the gold standard method to produce a straight line calibration curve that must pass through the origin. Now, a method that demonstrates good performance evaluation should have an R-square value above 0.95%. That is, it must be at least 95% accurate when compared with a gold standard method. Such method can then be adopted for analysis of a known analyte in a sample. Now, the second chromatographic technique in this presentation is high performance thin layer chromatography. High performance thin layer chromatography is an advanced form of thin layer chromatography that provide an enhanced separation, high sensitivity, higher resolution, higher reproducibility, and quantitative capabilities, as well as speed in analysis of compounds. It is the same as conventional thin layer chromatography, only that everything in high performance thin layer chromatography is aided by automated devices. Hence, it has the following features. Uh, it has better resolution. It has an automated sample applicator. Samples are applied automatically on the TLC plate and then developed. After development, the samples are the analyte in the samples are detected and quantified using an instrument called TLC scanner. The TLC scanner has the ability to scan the TLC plate at various wavelengths and quantitate the spot on the TLC plate. And hence, in this type of thin-layer chromatography, it does not require all the manual methods we discuss in the ordinary thin-layer chromatography. Everything is automated, and hence it has more application. It can be applied both qualitatively and quantitatively to detect and analyze in a sample. Hence, this technique is usually validated using the same protocol we discuss for ordinary thin-layer chromatographic validation. Sample system, solvent system developed, then when appropriate sample solvent system is achieved, the process is validated to ensure it meets up with the scientific standard. So this particular technique, it has various applications because of this sophisticated features. It is a very useful technique for creating fingerprint of microbial extracts to compare different strains of microorganisms. TLC, because it separates compounds based on their physical and chemical properties, so it is able to produce a unique pattern called fingerprint that can be compared between samples. So this allows researchers to quickly assess similarities and differences between microbial strains. For example, if these are microorganisms, based on this fingerprint, it can be seen that some of these microorganisms share some similarities and differences. Take, for example, microorganism C compared to microorganism E. These microorganisms are the same because their fingerprint is the same, meaning that they belong to the same strain. However, the microorganism in D, B, and A are different because they produce different fingerprinting. Now, this fingerprinting is very useful in chemotaxonomy. It is used as a valuable tool in chemotaxonomy to classify microorganisms based on their chemical composition. In addition, it can also be used like the ordinary thin-layer chromatography in all the other applications such as in pharmaceutical industry for quality control of drugs, identification of impurities, stability testing, and so on. It can also be used in food industries for analyzing food composition, detecting adulterants, and assessing food quality. It can be used in phytochemistry for identifying and quantifying plant-based compounds. It can be used in biotechnology for analyzing biomolecules and metabolites. It can also be used in environmental monitoring for assessing pollutant and contaminant in. Now, the third technique is high performance liquid chromatography. This technique is an analytical chemistry technique that is used to separate, identify, and quantify the component of a mixture uh, uh, under high pressure. So it works by passing the liquid sample under high pressure through a column that is packed with a stationary phase with different in which different components interact, leading to separation. The separated components are then detected and quantified using ultraviolet detector or photodiode array detector, which will send information into a computer software that can be detected on the screen. Now, this is a typical TLC. It has the separating system consisting of mobile phase, which can be 2 or more liquid samples that can be used and mixed at various ratios to provide separations of the analyte. Now, this particular system has an auto sampler through which the samples are applied, and then it also have a detector that detects the samples. So there are compartments. There is the solvent system, and then there is a compartment where samples are injected, and then there is a pump that pumps the solvent system mixed with the analyte and then send into the column. So when it's sent into the column, the separation takes place, and then the detector will now detect the component as they are being separated and eluted out from the column. And then the signal is sent to the computer software so that it can be visualized. Now, let us take a closer look at how high performance liquid chromatography is carried out. Usually the procedure is very simple, being automatic. Samples are injected through the injection port and then mixed with the mobile phase and being pumped through the separating column, which is usually C8 or C18 column that separate the component according to their polarity and sizes. And then as the component separate, they are being eluted out of the column, where they through gradient elution, where they pass through a detector that detect and quantify each of the separate component and visualize using a computer software. Now, let's take a closer look at the procedure. We have the HPLC system, and then these are the components. We have the solvent, which is the mobile phase. We have the pump. We have the samples being injected. We have the HPLC column detector, and then we have the monitor, the computer system that that can be used to visualize the detected component. Now, what happens is that the pump usually pump the solvent at high pressure and mix it with the samples that is being injected and pass them to the HPLC column at high pressure. So when they are passed to the HPLC column at high pressure, the separation takes place. As the samples enters through the HPLC, they are being separated according to their polarity and sizes. And as they are separated, they will now be eluted out of the pump and a separate component. As they are passing, they are passed through a detector. A detector that will now detect the component and send the information into a computer software, the HPLC software, which will now produce the response for each of the separate component in form of various peaks. So each particular peak represents a compound that has been separated in the column. Now, HPLC has several applications such as quantification of antibiotics, quantification of phytochemicals from plant, and so on. However, just like tiller chromatography, HPLC quantification or detection requires the presence of known component. That is to say, a known standard, a commercially available standard is used as a reference to detect and quantify an unknown component using the HPLC system. Just like in tiller chromatography, a standard or non-analyte is first analyzed using various solvent system until a very good separation is achieved. So the analysis is carried out at various concentration in order to have peak areas per concentration, various peak areas per each concentration. And then the peak areas versus concentration will now be used to produce a calibration curve. So the calibration curve of the standard will now be used to determine the concentration of an unknown analyte using the same solvent system. So if the analyte is present in a particular sample, when the same solvent system is used, it produces peaks at various points representing any of the analyte or all of the analyte, and the concentration of the unknown analyte will now be automatically calculated by the HPLC software using the calibration curve equation. And in addition, HPLC also requires validation, just like thin layer chromatography, to determine the linearity, limit of detection, limit of quantitation, precision, accuracy, and so on before it can be adopted for analysis of unknown compounds. Now, the last procedure covered in this presentation is gas chromatography mass spectrometry, which is an analytical technique that is used to separate components. GCMS combines gas chromatography separation with mass spectrometric structural analysis to separate individual components in volatile and semi-volatile compounds. The separation is usually based on volatility and mass of the component in the analyte. So unlike the HPLC, in which the separation is in liquid form, the separation in gas chromatography is in gaseous form, hence the name gas chromatography. Hence, a gas is used to serve as a mobile phase that carries the samples, and the samples are also volatilized into gaseous form, and then they are passed through the column by the carrier gas for the separation to take place. So GCMS has several applications. It can be applied to identify unknown metabolite, for example, phytochemical compounds from plants, metabolite from microorganisms, and so on. Now, the procedure of GCMS is simple and automatic, just like that of HPLC. It involves injection of the samples, ionizing the component of the samples, and then the fragment ions are now detected by a detector and sent into a computer for where they are visual, where the detected component are visualized. So it has a carrier gas which carries the sample and pass it across the column for separation to take place. Now let's look at the procedure. First, we have a carrier gas that carries the sample, which is injected into a capillary column. As the sample enters the capillary column, the GC oven is heated until the samples become volatilized. When it becomes vaporized, the sample is passed through a column. As it passes through the column is attached to mass spectrometer. The components are being separated in the column based on their volatility and masses. So usually there is an ionization source as the samples comes from the capillary tube. they are hit with the ionization source, which is high electric volt, that changes them into ions, and then the ionic fragment becomes separated in the column and then are being detected by a detector and sent to a monitor, to a computer, where they can be visualized and represented in form of peaks. So the peak area, each peak area represent an analyte. Each peak area represent a compound in an analyte, and therefore the area, the area, the percentage, the area correspond to the concentration of a particular analyte. Now, in order to uh, identify the component detected by the gas chromatography, a comparison is made between the ionization of each peak of each compound based on its peak. The ionization is being compared with that of the standard, and in this way the compound is being identified based on the ionization characteristics, while the peak area is used to calculate the relative percentage of the compound in the target analyte. So in conclusion, each technique has its own unique strengths. Thin layer chromatography is very simple and not much costly, and it can provide a fingerprint of a compound of interest in a given analyte. HPLC can be used to identify, to detect and quantify compounds in an analyte, and is more rapid, more automated compared to thin layer chromatography, and hence it is more reliable and it has higher speed compared to thin layer chromatography, although it is more expensive. GCMS is used to identify a known compound present in a sample. So selection of each method depends on the compound volatility. Non-volatile compounds are separated using thin layer chromatography or HPLC, while volatile compounds can be separated and identified using gas chromatography. However, a combination of two or more of this method provide a comprehensive profiling of microbial analysis. So with this, we come to the end of this presentation, and thank you for listening.